Now, the specific defense that we are going to learn now deals with immunity in general. Okay, so if we can understand the immunity first, like how we do the difference between multiple types of immunity, that we are going to learn first. Okay, and then we are going to go in detail of the specific difference. So immunity are two types. It's called acquired immunity and innate immunity. Now just pay attention to the name, innate immunity. That means the defense that you're born with. For example, easy example, skin. You're born with skin, you're born with stomach acid. So you already have that defense, you're born with it. It's called innate defense or innate immunity. Acquired means you get later on in your life, after your birth. So you get later on in your life. Now the question is, how do you get it? If you get the antibodies from another source that your body didn't make it, it's called passive. Otherwise, your body actively make it, it's called active. So one more time. So immunity, the one that you acquire it in your lifetime, it's called acquired immunity. The immunity that you're born with it, it's called innate immunity. So innate, you have no role, you're just born with that. But acquired, that's your role. So when you say acquired immunity, you have two types, active and passive. So active, passive, active acquired immunity, passive acquired immunity. Passive means somebody gave you the antibodies. And active means your body produced that antibody. Now, then the second question comes, like, who gave you? If you got it from your mother after your birth, that's so-called natural passive immunity, because in mammals, we know that mothers take care of their offspring. So if that antibody transfer across the placenta or via the breast milk, that is basically natural way of acquiring some antibody. That is why breast milk is so important, because it has a lot of antibody that's given to the children, like their next generation offspring. So that's natural passive. Induced passive means when you are sick, if you have a health issues, you are admitted in the hospital now, healthcare professional giving you antibody directly. They're injecting the antibody directly. So those antibody, they either manufactured from another source or they got it from another human being. So if I can isolate all the antibody that one person from one person who had the COVID-19 virus, so I can get all the antibody. If I can manage to have enough antibody, I can save another person because I can inject that antibody to another person. The second person is so-called induced passive immunity, meaning he or she is given the antibody. On the other hand, active means your body makes it. Now the question is how to do it. So one is when the healthcare professionals ask you to make the antibody. What does that mean? That means when healthcare professionals trigger an immune response in you, meaning vaccination. So they administer a little bit exposure or attenuated virus or bacteria or genetically made bacteria or heat-killed bacteria or virus. If they inject in your body will think that's an antigen, and they are, and your body will develop a lot of antibody to prevent from any kind of homeostasis imbalance. And then not only they made it, this is the beauty of the antibody, not only they will make it, but also they will memorize the incident. So next time, that is the purpose of vaccination. So they give you a little quantity, they give you attenuated, they give you a genetically modified, they give you a dead virus or bacteria, so that that your body knows then that this is my enemy. So they're ready to make the weapon against those enemies. So they're ready. So the second time, if you get infected by them, you can attack them right back. Okay, so that's vaccination. That's the purpose of vaccination. That's called induced active immunity. Means your active immunity uninduced. And the finally, naturally acquired immunity, that's basically you're on your own. Meaning you are getting infected by virus and the bacteria like antigens or any toxic elements in the those which are called antigen. So antigen that triggered the immune response from the environment, and you are on your own. If you can make it, fine. If you don't make it, you are dead. That's it. So that part is like you just actively makes, and your body can actively makes antibody depending on what kind of antigen they come across in their life. So that's basically it. Okay. So this is how we study the immunity. So right now in this particular chapter, we are going to talk about this, like how do we get our natural acquired immunity? Like how do we make antibodies? How do we kill the foreign element or antigen that from the environment comes to our body? To understand the immunity a little bit more, we have to understand science of the antibody a little bit more. Okay, the structure of the antibody I'm going to mention later on, but what they are actually, they're very specific. So that's their properties, like specificity, meaning they are going to find a specific antigen and they're going to just kill that. So if an antibody is against COVID-19, they cannot kill another virus. They cannot kill pox virus. Or if an antibody is against for hepatitis B, they cannot kill hepatitis C. I mean, they are very, very specific. Okay, so antibodies are specific to one antigen. Versatile, meaning your body system is so versatile, your immune system so versatile, they can combat and they can confront any antigen at any time. So you have a really versatile immune system. They can make any antibody. But then the question is like, why we're suffering from virus? The reason we're suffering is about the time. They can do it. That doesn't mean they do it next day. So depending on your body homeostasis, your body's condition, most people can do it, but there are few unfortunate people. They can't do it on time. You will be, but it takes time. It's like locksmith. You go to them and you ask to make a key for a lock. They will do it. Depending on the complexity of the lock, they can take anywhere from like 5 minutes to 5 hours or maybe 5 days. So if you do it quick, you are fine. So they are versatile. They are versatile means they can make anything depending on what kind of antigen you dare come across. Memory, meaning they memorize, it remembers the antigen. So once you get that antigen in your body fight back against it and they memorize them. So next time, they won't take 5 days, they take 5 minutes. So if you make a key from one lock, if you take it to the second time to the locksmith, if he or she saved that data, they can make it in 2 minutes. They don't need any 5 days anymore. Same thing, same concept. And final one is tolerance. Very interesting concept, although the detail of which is not in your syllabus. Tolerance means our immune system, they make antibody, which is true, but they don't make antibody against us, meaning against themselves. So they understand, there is 2 words they use. They understand self and non-self. So they own to make it, and that is applicable not only to the body, that is applicable to the day-to-day material as well. So for example, for most people, is there an exception? Yes. But for most people, the body does not make antibody against coffee or tea or most food, because those are the material that goes to your body every day. So they can't be an antigen because your body understands that's a tolerance. So they don't make antibody against kidney cell. They don't make antibody against any other part of your body, but sometimes they do. Those are so-called autoimmune disease. So when you hear the terminology autoimmune disease, it means your immune system by mistake trigger in such a way they are killing their own parts. They're killing their own body system. That's what happened in autoimmune system. So some people autoimmune, for example, like thyroid gland. So they're going to attack the thyroid gland for no reason. Okay. So lupus is an example of an autoimmune disease. So there are many of them nowadays, we know. Okay, so that's the properties of these antibodies and immunity. Okay. Now the question is, so how it works? How does these things works? How your immune system responds to this. So in order to understand that, again, this part of the chapter requires a true understanding. And I can guarantee one thing, if you do understand, when you read, it's going to be like a piece of cake. But if you don't understand, this is a very hard part of this particular area. Okay, so pay 100% attention to this. So now there are two kinds of cells in a lymphocyte group, the T cells and the B cells. NK cells, we learned already, they're involved in non-specific defense. Here we are going to focus on T and the B cells. Why it's called T cells again? Because it is called T cells. The reason is it's a thymus-driven cell. So the gland thymus participate in that process of maturing T cells. Why it's called B cells? Because it's a bone marrow-driven cell. Excuse me. So in the bone marrow, they develop, they're born, and then they develop. Now, how these two cells react differently? So in short, like it's say, if one antigen goes in the body, if T cells recognize them, they attack the antigen and they kill them most of the time. There are some issues that they cannot destroy those parts. What they do, they ask the B cell to destroy that. And B cell, when they mature, we call that plasma cell. The B cell, when mature, they produce antibody. So in other words, let's in a simple word is this. So T cells, what they do, they attack. So let's say, forget about T cells, B cells. So let's say there's a army group, group of soldiers that are fighting for the country. So the army normally, what they do, just in a very simple sense, they will kill the enemy. So in army, they see the enemy, they recognize them, and then they kill the enemy. But sometimes nowadays we know, even when they kill the enemy, the danger is not over. Because sometimes, for example, just an example, let's say enemy has a toxic product attached to their body, or enemy has a bomb attached to their body. So now you shoot the enemy, you kill the enemy. Okay, your first goal is over. Now the enemy has a bomb attached to it. If that blast, that would cause a lot of damage. So now the person who shot that enemy doesn't know how to take care of the bomb. So T cell, they kill the antigen, but doesn't know how to take care of the toxin, which is another antigen. So what they do, they call the B cell, they call the bomb squad. And the bomb squad come and then defuse the bomb. So the B cell come, diffuse those toxins. The B cell produce antibody, antibody diffuse the toxin. So that's how actually body works. So you're going to break down that step later on to understand exactly what step they works. So when there is a specific, but everything specific, because we have to understand, we have to recognize that particular enemy. So we have bacteria and viruses. We recognize them. We recognize that if it is, let's say it's COVID-19, we recognize them. Or it's a pox virus, we recognize them. Or if it is a particular streptococcus bacteria, we recognize them. So once we recognize them, either we can use the cell-mediated immunity, meaning T cells kill them directly, or we can say B cell-mediated immunity, meaning antibody destroy the antigen. So when T cells get activated, the direct physical and chemical attack, meaning they're directly shooting them. Let's go back to our own example. And they could not diffuse the bomb, then B cells produce antibody, they're killing the antigen. That can also happen. So there are cell-mediated defense. We call them cell-mediated because the cell is participating directly. And we call antibody-mediated defense, or where the antibodies, so some book use the term humoral immunity. Humor means the liquid. So that's where the antibody is involved. So one is cell-mediated, one is antibody-mediated. Cell-mediated done by T cells, antibody is done by B cells, okay? Now, how they do it? That's the question. So let's focus first on the cell-mediated one, like how exactly do it. In order to understand that, we first need to understand there are not one type, multiple type of T cells. So same thing like, when you say US Army, there is not one type, there are multiple type of Marine, the Delta Force, the Navy, the Air Force. So we have different type of people. Their job is slightly different, but the goal is same. The goal is to defend, defeat the enemy and defend your body, defend your country. The goal is same, but the attack is different way. Same thing here. So T cells, some T cells are called cytotoxic T cells. That means they're toxic to the other cells. They're toxic to the other cells. We call them TC cells. What they do normally, they attack the foreign cells regularly. Helper T cells, the name is the answer. They help in the process. So they activate other T cells and B cells. And suppressor T cells, that's very interesting to know. Some book also call it regulatory T cells, Treg or TS. Suppressor T cells is basically, remember, it's an army. You don't want to stimulate the army for no reason, because their training is to kill. So you do not want to trigger a killing machine in your body without understanding the consequence, because the killing is a non-reversible process. It's like an irreversible process. You cannot kill somebody and say, hey, oh, I made a mistake. So there is no room for mistake. So we need to make sure that everything is perfect. So suppressor T cells, what they do, they suppress the activity of these cells. So they have a control on it, a regulation on this. But the regulation part of the control mechanism, it's not in your syllabus. I'm going to go only with these two from now, cytotoxic and helper T cells. So how they work, how they do. So this is the step. So if you understand Call of Duty or how army works, you understand this. So basically, you don't have to go for detail of this. So the army is fighting against the enemy. So what is the first step? The first step is you got to recognize who is the enemy. That's the first step. So you have to recognize who is the enemy. And once you recognize, then only you can kill it. Otherwise you can't, because you don't know who the enemy is. Now the question is, how do you recognize enemy? So there are some cells that help you to recognize. There are some people, just giving an example here, like there are some people, let's say in FBI or CIA or the intelligence community that we have, they recognize those enemies. And they're telling the army, hey, this is the enemy. That's what the antigen presentation word is coming. So those cells are helping the army and telling them, hey, this is the enemy, kill them. So they're telling them. So that's why the word antigen presentation is. So this is very interesting, the antigen presentation part, because all our body cells, they can be part of it. So all our body cells from their DNA, the genetic material, those which has the nucleus, obviously, if you don't have the nucleus, you don't have this. So blood cell, mature red blood cell does not participate in this. But those which are nucleated cells, they produce a type of protein called MHC proteins. MHC proteins are major histocompatibility complex protein. So they are coded by the genes of MHC. So in your DNA, you have MHC genes that makes MHC proteins. All cells have it. Now, depending on what kind of MHC, we divide the cell in two groups. Class 1 MHC, class 2 MHC. So there are two groups of MHC protein. Class 1 protein found in all nucleated cells, all of them have it. Class 2 found on so-called antigen-presenting cells and lymphocytes only. Lymphocyte is also part of antigen-presenting cells, meaning those cells, when I'll give you examples, you'll understand they are so-called trained cells. So class 1 and class 2, let's put it this way. So for example, if you consider me, like I am not trained in fighting, so I don't know how to fight. So if, imagine this, a bad guy comes to my house and attack me, the choice I have is to call for help, or cry or run. I don't have any other choice because I'm not trained fighter. So yes, if I cry or run, both cases there's a chance I'm going to die. So the slimmest chance I have is to call for help. Our body cells, some of them does exactly like me. So they are like a professional group of body cells. They can't fight. But the other people in society, for example, the people who are in army, people who are police officers, people who are in CIA, FBI, all these people, they're trained. So if a bad guy attacks them, there is a chance that the bad guy will be killed. So in our body, exactly same thing happened. So when a bad guy attacked the first group, let's see what happened. And then we're going to see what happened, bad guy attack. So let's learn first that when a bad guy attacked me, what happened. Then we're going to learn what happened, bad guy attack and police officer's house. So let's look at this one. So what happened is, as I mentioned earlier, that every nucleated cell, they have MHC proteins. So when a virus injecting their DNA, it could be viral or bacterial infection, any one of them. So when they infect the body, infect the host cells, what host cell does, that MHC protein class 1 they have, the class 1 MHC protein capture one of those viral or bacterial particles. So one of those particles, like abnormal peptides, the portion of the protein, they take that protein, bind with that, and they show it outside on the membrane. So basically, this is what's happening. So as soon as a bad guy attack my household, some member of my house is going to try to call 911. That's the 911 call. So I'm putting a flag outside in my house, just literally, and telling the people like a police and the other people who can save, who can fight, that, hey guys, I am infected. So that's what the cell does. Right here, they put the flag out there telling the neighboring cells that, hey, I am infected, kill me. Meaning, in human body, slightly different than our own social life. In our own social life, I'm asking for the help. I'm not telling them, hey, come and kill me, but I'm asking for the help. But here, by killing themselves, they're saving the neighborhood. So the cell, what they do, because they know that they are infected, they are not going to make it. So they are already infected, but they can save the body, which is our goal. So they can save the neighborhood, they can save the tissue. So what they do, so they, outside the membrane, they put a flag out there. So this is class 1 protein. That class 1 protein holding abnormal polypeptide with a sign saying, hey, I am infected. But class 2 protein, they are slightly different. When they attack, this is a police officer house. So when an antigen attack them, virus or bacteria, they kill them. They destroy the antigens. See that they have lysosome, all other mechanism of defense, they can kill them. But now comes the problem. Remember the discussion I had, Let's say they kill the bad guy, but then the police officer realizes, oh, there is a bomb here. And that particular officer may not be trained with how to defuse the bomb. So they will also call for help, but they don't want to kill them. They said, hey, defuse the bomb. So to help is two different types. So the cell before it, they said, hey, I am infected, kill me, save the neighborhood. And this cell saying, okay, I killed the bad guy, just diffuse this antigen or diffuse this bomb. So this is the antigen they're holding. So class 1 protein sending a different signal than class 2. Class 1 protein signal is kill me, I am infected. Class 2 protein signal is this is a bomb, diffuse it. So that's the difference. So we have exactly these two classes of cells in our body. Now what happened? So now those defense people come, those the army, those the police, the all defense people, they come. There are many of them. They are different types. Then when scientists discovered them, they found out those T lymphocyte or T cells, their membranes contain a lot of markers. We call them CD marker. CD is a short form of cluster of differentiation molecules, CD markers. So we name them like 3, 4, 5, 6, and so on. So there are many CD markers to discover. You just have to remember 2 CD marker, not even 3, 2 CD marker, CD8 and CD4. These are like antennas on the cop cars. You know, there are different types of antennas. Some antennas find this kind, some antennas find the other kind. So the same thing here. We have different types of marker. Which one you have to remember is this? CD8, they are on the cytotoxic and suppressor T cells. So we are interested in right here, cytotoxic. And CD4 is on the helper T cells. Okay, so CD8, those are cytotoxic T cells. CD4, they're helper T cells. Now the very important part. CD8 recognize class 1 protein. CD4 recognize class 2 protein. So when we say that this is, I am infected, this will be recognized by CD8 marker. When I say this is a bomb, this will be recognized by CD4 marker. So this is helper T cell recognize them, and here cytotoxic T cell recognize them. So let's see what happened when cytotoxic T cells recognize them. So CD8 marker containing cytotoxic T cells, what happened when they recognize them? This is what happened when they recognize them. So when they recognize them, very important steps. So this is the infected cells. That's the MHC class 1 protein. They're showing that protein right here. That's a flag. They're saying, hey, I am infected. This is the CD T cells. So the T cell came. Now, this is how even our society works. They just cannot, the police officer is not going to come like, or the SWAT team showed up in your house and then start shooting. That's not going to happen. They want to first find out what is the true story. Because remember, killing is an irreversible process. You kill and then you cannot just say, hey, I am sorry. That's not going to solve the problem. So what they do, they want to make sure it is a real case situation. Same thing in our body. The T cell, once they recognize that, they just don't shoot them and kill them. They want to make sure that yes, that cell is infected. So there is a step called co-stimulation. So the CD8 marker, they're going to send, this is a CD8 marker, they're going to send a lot of signaling molecules, signal transduction, that's in upper level classes you're going to learn. They're going to send the signaling molecule to make sure that it is infected. Once they co-stimulate and make sure that it's infected, then now the T cell undergoes cell division. Because remember, it's not fight against 1:1, it's fight million versus million. So we know, we already, when it is microbiology, it's not like one cell is infected. Let's say your bacteria infect, let's say 1 million cells. So now your T cells undergo cell division. Now here's another beauty. As soon as they're dividing cells, they're making a lot of active cells, but some of them keep it inactive as a memory cell. So some T cell they become memory cell. Some of them, they become memory cell. That's the one that can recognize if the same bacteria or virus come or the same antigen showed up later on, then we don't need this co-stimulation step because we know they're bad. So those memory cells jump on the in the action later on, not this infection, later on in subsequent infections. So the rest is very simple. Cytotoxic T cells, once they're active, they're ready to kill. Question, how they kill. Remember, you can kill an enemy with a gun, with knife, with poison. You can tell the enemy, hey, you either die yourself or I'm going to kill you. So many things, same thing here. So T cells, they release lymphotoxin. So lymphotoxin destroy the other cell metabolism. So the bacteria or virus will die. They release cytokines, telling bacteria or virus undergo suicide. Apoptosis means cell death, programmed cell death or suicidal attempt. So they're telling bacteria, either you commit suicide or I'm going to kill. So that's what they do. And they release perforin, that's the protein, creates perforation or the hole on the membrane. So the destruction of the cell membrane and they can die. Bottom line is, no matter what pathway they use, they're going to kill the target cell. That is what is called cell-mediated immunity. So when class 1 protein captured the antigen, call for cytotoxic T cells. Cytotoxic T cells appear there, co-stimulate, make sure they are the infected one, undergo cell division, keep some memory cell for future use, and then attack them via lymphotoxin release, cytokine release, or perforin release. Now, what happened to the other one, the class 2? So I'm going to teach you this one right here. So class 2 is recognized by CD4. That's another class of T cells. That's called helper T cells. They recognize them. They say, oh, yes, that's the bomb. But I don't know how to do it, but I know the group who can do it. So what they do, they undergo cell division. They keep some memory so that they can remember this bomb for future use. And then they call for help. Who is the help? B cells. In the body, B cells are sensitized. In the body, B cells are sensitized. This is called so-called sensitized B cell, meaning they're ready to fight. They're trained, ready to fight. Okay, they have the bomb suit in. So now helper T cells, they come and ask the sensitized B cell, hey, there is a bomb, can you defuse it? And then that sensitized B cells, then before normally what they are trained to destroy the bomb or defuse the bomb, but they have to wear a bomb suit and stuff and then become the real person who does the job. Same thing here. B cell is sensitized, they know how to do it, but then they differentiate. So they activate B cells. Activated B cells, we call them plasma cells. They are literally a bigger one. So this is not a different cell. This is when B cells get differentiated and activated, we call that plasma cells. Plasma cells then release that specific antibody, which can go and destroy that specific antigen, meaning which can go and destroy that specific bomb. So for every antigen, we have a different type of antibody. So body does not produce one type of antibody, body produces thousands of types of antibody depending on what is the antigen. So these B cells are also specific. So the B cells will produce the antibody against COVID-19 is not the same B cells who's going to produce antibody against pox virus. There's another group of B cells. So we have lots of memory B cells depending on that vaccination process or the one that you acquire yourself. So, and they can identify and kill them. That is how they work. Now, a little bit of the antibody. Antibody is a protein molecule. It is a protein molecule, consists of like polypeptide chain. We have 4 chain of antibody. In one antibody, we have 4 chain, like 2 heavy chain and 2 light chain. Heavy chain means it's a long chain, molecular weight is heavy, and light means molecular weight is less. So that's why they appear light. But in both chains, so this is the heavy chain, it's a long molecule. So from right here to here, this is also heavy chain from here to here. They have a lot of bends and stuff like that. So, and this is the light chain. That's a one light chain, another light chain. So you have two light chain and two heavy chain. They are also bound together. So it's like a one big molecule. They are attached. The light chain is attached to the heavy chain, and heavy chains are attached by all these disulfide bridges. So they create one quaternary structure. It looks like a letter Y, and it's an antibody. All the antibody looks same. Their look is same. It's like the key, typical key looks same, but each key is for different lock because they have a little bit different binding site. So what is the binding site? The top part is called binding site for antigen. Now, if you pay attention to the antibody structure, the top part of both heavy and light chain, the top part of both heavy and light chain are so-called variable segment. That means in different antibody, that's the one that varies. The bottom part for the light and the heavy chain, which is in this picture solid color, and this is the shaded color. So the solid color part is called constant segment, the constant part. So if you just imagine this, I'll give an example. Let's say there are 5 car companies make cars, 5 car companies like Toyota, Honda, BMW, Mercedes, and Nissan. Okay, so Toyota, Honda, BMW, Mercedes, Nissan, these are the 5 car companies. Now, if you take a Toyota car keys, that key is not going to open all the Toyota. They can open one, like your car maybe. If you take a Honda car keys, they cannot open all the Hondas. They can open my Honda. So in each individual, you can have thousands variety. Same thing in our case. Based on the constant region, based on the solid part of heavy and light, we have 5 types of antibody. 5 types of antibody just based on that constant segment. Toyota, Honda, BMW, Mercedes, and Nissan. Same thing here, 5 types. We call them antibody like GEDMA. This letter came from a different, so like a gamma structure, alpha structure, mu structure. So the Greek word, but here you remember GEDMA. GEDMA, GEDMA. I just made up that. So it's like 5 different classes of antibody, GEDMA. Now, antibodies are immunoglobulins, the protein called immunoglobulins. They're globular proteins, so we call them IgG, IgE, IgD, IgM, IgA. That's why we call them like GEDMA, IgG, IgE, IgD, IgM, IgA. 5 classes. Each class can be 1,000 variety. So in a lifetime, like you can produce, let's say 20,000 antibody. So you may have 10,000 IgG, 5,000 IgM, and so on, depending on what kind of antigen attacked you. So how do you make different type of IgG? Just changing the variable region, changing the top part of the key. The bottom part remains same. So if you take 2 Honda keys of the same year, most likely the bottom part will look same, but the top part is different. That's the variable part. So the variable part of light and heavy chains are different. And the bottom part, the constant part, are the same. And that is why we create multiple different type of immunoglobulin molecules, which keep yourself safe. Different molecules has a little bit different functions. So overall, what they do that you need to know. You do not need to memorize what IgG does, what IgE does, what IgD does individually. They have different jobs, but you don't. You need to know overall they do. So overall, what their main function is, neutralize the antigen. Neutralize the antigen. So the immunoglobulins, their main job to bind with the antigen and neutralize them. So when they bind, we call that phenomena like agglutination or precipitation. So it is very similar that you learn in blood chapter when anti-A antibody bind with the blood and a blood type A, and they coagulate the blood. They agglutinate the blood. That same thing. They can also activate the complement system. We know part of the antigen function, antibodies function, is to trigger the complement classical pathway. So if you forgot, go back to read the classical pathway, that when C1 complement protein bind to the antibody, that's what it's written. attraction of phagocytes. So antibody binding can also attract the phagocyte. Remember, when we attack the enemy, we don't attack 1:1. We attack like our Army, Navy, Air Force, everybody goes together. That's exactly. This is called this should be opsonization, SO, not OPC, opsonization. Opsonization is like facilitating the attack. That's what the word is. And stimulation of inflammation. Inflammation means the body's response to the injury. So that's what the stimulus. Prevention of adhesion, because the bacteria and virus, what they want to do, they want to adhere to a host cell before they inject their DNA or before they enter into the cell. Antibody prevent that, because the antibody is binding the bacteria. Now bacteria or the virus cannot bind to the host cell. So if they cannot bind to the host cells, they cannot inject their DNA, they cannot infect the host cells. That is how they actually work. So these are the 5 class: IgG, IgE, IgD, IgM, and IgA. So that is how antibody, that is how the specific defense is working. So in summary, the specific defense can go with the T cell mediated antibody, or we can say cell mediated immunity, or we can say humoral or antibody mediated immunity. So in order to kill, we got to present the antigen. How do we do it? We use the 2 classes of MHC protein, class 1 and class 2. Class 1, which can which it's in all nucleated cells. So class 1 has all nucleated cells that already have the class 1 protein. When a foreign antigen attack them, class 1 protein capture one of their abnormal peptides, show it on the membrane so that they can attract the T cells or cytotoxic T cells, because they are attracted by those cells who has CD8 marker. So class 1 is attracted by CD8 marker. So CD8 marker carried by cytotoxic T cell, they come and they recognize, they co-stimulate, they divide, then they attack and keep some memory. So class 2 is they destroy the bacteria or virus, because those are antigen presenting cells such as macrophages, lymphocytes. So they're fighter. So they destroy them, and now they're holding some of the antigen, because antigen could be poisonous, antigen could be toxic, they can be harmful for the body homeostasis. So they are recognized by CD4, which carried by helper T cells. Then helper T cells ask help from the B cells. Those are sensitized B cells, meaning they're ready, they're trained, they are ready to make lots of antibody. And then they become plasma cell, which finally produce the antibody.